I'm delighted today to be able to introduce Janko Gospocic for the talk that he's going to give. So Janko is Croatian and was born and raised in Croatia, and then did his foundational scientific work at the University of Zagreb. And after that, he went to the European Molecular Biology Laboratory and did his PhD with Paul Heppenstall. After that, actually, for what would be equivalent to our postdoc training, he went to work with Roberto Bonacio, and he spent half of his time at the University of Pennsylvania and the half of his time at the University of Freiburg, doing foundational work with an interesting ant colony. He was recruited out of his postdoc to come to UT Southwestern with an Endowed Scholar Award. And since getting that Endowed Scholar Award, he also has added to it from NIH and R34, one of their new grants called Maximizing Investigator Research Award. Janko's work focuses on a particular kind of ant colony. I'll give it a try to pronounce it, the Harpaginosaltator, ants. You can say it better. He's using these ants, which are genetically similar ant colony, to study the epigenetic changes of behaviors that are specific to social behaviors. These are behaviors that are associated with caste phenotypes. The model system will allow Janko to ask questions like how social interactions, how aggression, how social defeat induce epigenetic changes for long-lasting behavioral phenotypes. And these are behavioral phenotypes that we care about and that we use within psychiatry. His model system is fascinating, and there's nobody that can talk about it better than Janko Gospocic.
Thank you, Carol, for a kind introduction. So today I will talk about the questions that drive the science in my lab, and that's how the epigenetic pathways regulate behavior and aging. And I will introduce our unique model system that hopefully will help us to understand these pathways a little bit better. So in general, what is my lab is interested in is this correlation between epigenetic regulation of transcription, certain neuronal types, and the behavior. And these interactions are sort of two-way stream, meaning that the changes in transcription can lead to changes in the behavior, but also social interactions, especially negative ones like social defeat, can be very powerful force to change the chromatin landscapes and the transcription and the further behaviors. So in our lab, in combination with the more classical model systems of fly and mouse, we use very unique ant model systems. So why ants in the context of epigenetic studies? So here on this picture, you can see these three individuals with very different morphology. They are females of the same species, one of the classical ant species called Camponotus floridanus. In this species, the colonies consist of a single reproductive queen and thousands of sterile workers that can be major or minors. So we see development of these castes, we see very strong parallels with the stem cell differentiation. In the same way, depending on the external cues that the embryos and the larvae get during the development, the female embryos will become one of the female caste, or the queen, or one of the sterile workers. And not only that morphology and ovary activation is different between these castes, there are many other physiological phenotypes, and one of them most striking is different aging rate. So in this species, queen can live more than 25 years in the captivity, where the workers live up to 1 year. Once when they're set in this developmental trajectory, they cannot switch caste anymore. And these trajectories are completely regulated on the epigenetics level. So, for example, if we administrate the HDAC inhibitor directly by injecting it in the brain of the major at the critical period, we can actually switch the major behavior that it looks more similar to minor. So this division of the labor proved to be the most successful. It proved to be very successful, and the ants are the most abundant group of insects in the world. Today, there is more ants by biomass than humans, and there are more ant species than all the mammals and birds combined. But it has also downsides. So if this queen dies, the whole colony dies out because they cannot replace the main reproductive. So main species that we work in our lab is called Harpegnathus saltator. It's a very unique species. There is only single worker caste, but all the phenotypes that we see in the classical ant species are there. So the workers are very aggressive with intruders. They are sterile. The queens live much longer than workers. So in this species, the queen lives more than 5 years, where the workers live only around 200 days. But what is unique about this species, if the queen dies or it's removed from the colony, that's not the end of the colony. So if we make parallels of stem cells with the classical species, then I like to say that this sort of species is more like induced pluripotent stem cells. So when the queen is removed from the colony, the workers know that queen is not there anymore, and they start this ritual dancing tournament, and whoever wins that dancing tournament becomes a new queen. We call them gamergates in this case, and for the rest of the talk, I will refer to them as gamergates. So not only do they change behavior and activate the ovaries, they also change metabolism, they change aging rate. Now they live much longer than they would be as a queen. And not only that this works in one direction, it works also in the opposite direction. So if in the lab we put the old queen back, she's usually more dominant. Workers know that the old queen is there. So they pick up the new queen. She goes in this pupal position. They carry her around the colony. They put her outside of the nest. She perceives that as a social defeat and switches back to the worker. Again, completely rewires the brain, changes behavior, changes metabolism, and now she lives much shorter than she would be if she would stay as a gamergate. So in my lab, like I said, we are interested in two questions. And first question is how the epigenetic pathways regulate these social hierarchies. During my postdoc, I showed that steroid hormone signaling plays a very important role in regulating who is the worker and who is the gamergate. There is actually interplay between two steroid hormones, one that is associated with worker type behavior and the other with gamergate type behavior. Not only, we also discovered a very unique transcriptional factor called Krupel homolog 1. In this transcriptional factor is actually downstream of both hormonal pathways, and it acts sort of like a barrier, meaning that in the workers it's bound to the genes that are usually expressed in the gamergates, and vice versa. In gamergates, it represses genes that are important for the worker type behavior. So this slide is just because I like to show more ant videos. So this is one of the behavior assays that it's hunting that we do in this ant species. If you look this red ant, labeled ant, you will see that it will start hunting crickets in the tube. So this is a typical worker behavior, and gamergates don't do this type of behavior. So if we knock down this transcriptional repressor in the brain of the workers, they stop hunting and they behave more like agglomerates. And if we knock it down in the agglomerates, they start hunting and they behave more as the workers. We looked extensively transcription and the binding patterns of the chromatin, and it really is one of the main regulator of the Cas identity. So it acts actually with in the complexes with the chromatin transcriptional repressors like HDAC complexes. So not only that one pathway is important for regulating these transitions in the ends between the worker and gamergates and vice versa. If we looked at the RNA-seq, we saw multiple chromatin modifiers that actually change very early during the switch. Here are the chromatin modifiers that actually homologs were described in the GWAS studies as the potential targets for the autism-like disorders. And that's our next step, that we will focus on a couple of these chromatin modifiers. Some of them function is very well described in the neurons, but some of them, the function is completely unknown. The second question that what we are asking in our lab is how the epigenetic mechanisms regulate these different aging rates. How I mentioned is workers in this species live only for 200 days, and the gamergates live over 6 years. So if we have a worker that it's already at the end of their lifespan, let's say day 180, and it already deteriorates, if we cause social weaning and switch it to the gamergate state, they can completely rejuvenate. And they rejuvenate in a very interesting way because we don't get new neurons that would replace all the damaged neurons. We rather actually get completely epigenetic restructuring that cells look much younger than their real age. The only similar thing that happens that I know is with induced pluripotent stem cells. If you take the fibroblasts from the old patients, make them stem cells, and then differentiate them in the fibroblasts, they look much younger than their actual age. And that's sort of basis of all cell therapy. But like you, majority of you know, neurons take a really long time to mature. So we are hoping that we will actually find the pathways that in the end, they can rejuvenate these sort of neurons, but neurons still stay neurons. They don't go through the differentiation stage. And one of the first sort of pathways that we are focusing is on the heterochromatin. So during the normal aging, we see in all the organisms loss of heterochromatin marks like K9 trimethyl and K27 trimethyl. And then with rejuvenation, in this case, we see restructuring of these marks. We are small lab. We started year and a little bit ago. So we are looking for the hiring students and postdocs. So if someone is interested, can contact me. I would like to thank all my previous and current collaborators, and I want to give a special thanks to everyone in the Department of Psychiatry and Green Center that actually helped me in the last year to establish my lab here. Thank you, and I'm ready for your questions.